I invite Reverend Father Reggie Picurian, Principal St. Berchmans College, to welcome the gathering.
Thank you, Nidin sir. Distinguished guest of honor, Sir Richard John Roberts, Professor P.J. Thomas, convener of Berchmans Erudite Lecture Series, esteemed participants from all over the globe, good evening to all. I am immensely proud and happy to welcome you all to the 5th Berchmans Erudite Lecture, which is taking place in the centenary year of this college. Berchmans Erudite Lecture Series, we call it BELS, was instituted in 2012 with the generous financial contribution from the faculty of our college to mark the 90th birth year of our college. We call it Navadi. BELS aims to invite Nobel laureates to the campus for lectures and interactions with the faculty, students, and the public. Today, we are extremely privileged to have with us a great scientist, Nobel laureate, Sir Richard John Roberts. Sir Roberts will be introduced to you in detail shortly. He was awarded the Nobel Prize for Physiology or Medicine in 1993 for the discovery of architecture of the genes. He proved that the genes could contain coding regions called exons and non-coding regions called introns. Later, his discovery proved extremely beneficial in various fields of life sciences, especially in genetic engineering and in the annotation of genome sequences. Respected Sir Roberts, with lots of gratitude and respect, I welcome you wholeheartedly to this august gathering. On this graceful occasion, I cherish the love and affection St. Berchmans College splendidly received from the Nobel laureates, late Professor Ishi Negishi, who inaugurated this talk series in 2016. I also remember with gratitude the generous support we are continuously receiving from the former speakers, Nobel laureates, Professor Ada E. Onag, Sir Anthony James Leggett, and Professor Martin Shalfie. Our manager, Reverend Dr. Thomas Padiyat, who does not need any formal introduction at all. He is with us in all our endeavors and motivates us to do the task with utmost sincerity and commitment. Dear Father Manager, I proudly welcome you to this function. President of this meeting, Professor P.J. Thomas, convener of BELS, is the man behind the successful continuation of this lecture series. In fact, the actualization of the 5th edition of BELS is the fruit of the continuous hard work of Professor P.J. Thomas. Dear sir, warm welcome to you. Again, I would like to welcome all my dear colleagues behind this program, especially Mr. Ajay Jose, Secretary BELS, and other committee members of Berchmans Erudite Lecture Series, who have constantly extended their support to this great event. And with gratitude, I remember the efforts they put to conduct this event. Hearty welcome to you all. I am extremely happy to learn that participants from different parts of the globe have joined BELS through the Zoom platform. On behalf of the BERCHMANS College community, I extend you all a hearty welcome. I also would like to welcome our former teachers and students from the different departments who are part of this institution, and I welcome them all in my modest words. Once again, I welcome you all to this program and hope that you will have a very exciting and enriching academic session. Wishing you all success to this program. Wishing all the success to this program, I conclude. Thank you.
Thank you, Father. Thank you for your words of welcome. I request Professor P.J. Thomas, Associate Professor of English and Convener BELS, to introduce the speaker.
Thank you, Professor Nitin. Am I audible?
Sir, you are audible. Yeah.
All right. Thank you, Professor Nitin, and thank you, Father Principal, for that warm welcome. Good morning, Sir Professor Richard Roberts, and good evening to all the distinguished participants of this Nobel lecture in the prestigious BERCHMANS Erudite Lecture Series instituted by the teachers of the college to mark the 90th year of its foundation. So I remember with gratitude all the former teachers who have contributed generously to this cause. I also remember with gratitude the former convener and the first convener, Professor A.J. Thomas, who is also there in the audience. Let me also welcome all of you to today's lecture by Professor Sir Richard John Roberts, the Nobel Laureate for Physiology of Medicine in the year 1993. Professor Roberts is a British biochemist and molecular biologist. While introducing him today, I am reminded of the words of a French philosopher, Bernard of Chartres, who said, I quote, "We are like dwarfs on the shoulders of giants so that we can see more than they and things at a greater distance, not by virtue of any sharpness of sight on our part or any physical distinction, but because we are carried high and raised up by their giant gaze." I am today introducing to you such a giant with a giant gaze. Professor Roberts' gigantic achievement is the discovery of the alternative splicing of genes and the identification of introns in eukaryotic DNA, which has had a profound impact on the study and applications of molecular biology. His research in this field resulted in a fundamental shift in the understanding of genetics. Professor Roberts is also a receiver of many honorary doctorates and winner of numerous awards, which includes Golden Plate Award of the American Academy of Achievement. He is also a fellow of the European Molecular Biology Organization and a member in the advisory board of Patient Innovation, a free venue for patients and caregivers of any disease to share their innovations. He is also the chairman of the Laureate Science Alliance, again a non-profit initiative supporting research worldwide. And he was knighted in 2008 because of his contributions to science. He's a strong advocate of genetically modified organisms in general, and particularly golden rice, which is a rich source of vitamin A, which in turn is a great blessing for children in sub-Saharan Africa and South Asia who suffer from vitamin A deficiency. A self-professed atheist, but with strong commitment to human goodness, Professor Roberts is a wonder. So with tremendous gratitude, but also with great trepidation, let me invite, in the name of all of you, Professor Roberts to give the 5th edition of the BERCHMANS Erudite Lecture. Thank you very much for accepting our invitation. Now over to you, Professor Roberts.
Okay, well, good evening, everybody, and thank you for those kind words. Let me see if I can share my screen with you. I'm going to talk today about bacteria. And I think for many of you, bacteria doesn't carry a particularly strong connotation. You tend to think of them as the causes of diseases. And it's true, they do cause some diseases, but the vast majority of them do not. The vast majority of them are essential for the health of this planet, and in particular for our health. I will take you on a quick journey through some of the bacteria that you may not have heard of, tell you a little bit about those that do cause disease, and then finally talk to you about how beneficial these are. I want to start by taking a quick look at the tree of life. The tree of life are the various organisms that we find on this planet, and all of these organisms evolved over about 3 billion years from something, we don't quite know what, that was almost certainly a single-celled organism of some sort. The two major phyla, bacteria and archaea, shown at the top, are all single-celled organisms, and they're probably much more closely related to the root of life, to the original organisms. And you'll see that there is great diversity there. Now, down at the bottom are the third group of organisms, the so-called eukarya, and these are all multi-celled organisms. And down at the bottom left, and just over to the right, just over to the left rather, you will see at the end of that branch of the tree of life, you'll find Homo, man, sitting next to Zea, which is maize, and then on down to many more. And so you can see that man is a long, long way from the bacteria. And yet man depends very heavily on the bacteria for everything that goes on within our bodies. Now, I want to start off by talking a little bit about an interesting organism. It's a marine organism, Annabinus St. John's, lives in the water. And it actually probably has more to do with our being here today than anybody realizes. And the reason is that Annabinus St. John's and its relatives were the organisms that first pumped oxygen into the Earth's atmosphere. They were anaerobic organisms, but they grew on the rocks that were present in the sea. And they slowly introduced oxygen into the atmosphere. And in the process, they almost wiped themselves out because they were anaerobes. But what they did was to form small circular bodies within these long strings of cells, and that could survive in an aerobic atmosphere. So these were, in a sense, precursors of the things that many other organisms have done, and that is to wipe out their environment, something that we are dangerously close to doing at the moment. I think we have to make sure that we do not wipe out the life on this earth. Now, another organism that you probably never come across before are the Mixobacteria. The Mixobacteria grow in forests, they grow on soil, they grow almost anywhere where you find plant life rotting. And they have these interesting mushroom bodies that they form, which allows them to expand and to allow the population to spread. They're absolutely essential for moving the nutrients that were present in plants back into the soil and eventually back onto the planet Earth. Here's a close-up of one of these fruiting bodies of the Mixobacteria. They're really rather pretty, but without a microscope, you really cannot see them. Another very interesting organism is called Aquaspherilum. It has a very interesting morphology. Within the bacterium, you can see a black line that runs towards the center of the cell and is in fact a magnet. And with this organ, this magnet, Aquaspherilum finds which way is north and which way is south. And it uses this in order to arrange the particles in a chain of Aquaspherilum along the geomagnetic lines. This organism was isolated from a water treatment plant, and they're everywhere. There are many, many of these. If you look anywhere where water is stored and open to the atmosphere, you find Aquaspherilum. Now, I want to take you from the rather cold water where Aquaspherilum grows into the hot vents. The hot vents are these essentially volcanic eruptions that take place underwater. And they produce hot black smokers. These are chimneys of magma, chimneys in which smoke erupts from the side of the Earth. They come out. This is where plate tectonics is important. And you can see in the slightly to the left of the center of this picture, the black smoke that is erupting. And when this comes out from the magma, the temperature is of the order of about 350, maybe higher, degrees centigrade. Very, very hot. But of course, because it's underwater, it quickly cools, and one has a gradient down to this group of organisms on the right. And these are tube worms that are growing at about 20 degrees. And so this heat gradient allows a variety of different forms of life to grow. And in fact, these vents typically will last for many, many years. Life will begin to accumulate on them, having been transferred from previous vents, will start growing. And in this way, we have life, new life forming within the under the surface of the oceans. Sorry. This is a picture of tube worms. These are the tube worms that you saw on the previous photograph. They are very interesting. They are essentially just tubes, and they have living within them bacteria. And the bacteria that live within them provide all of the nutrients that the tube worms, which are eukaryotes, they provide everything that they need in order to grow. Now let's go from hot water under the oceans, and let's go to hot water on the surface of the Earth. This is a picture from Yellowstone National Park, one of the most wonderful national parks that I've ever been to. If you ever come to the US, I strongly suggest a visit to Yellowstone if you have the opportunity. And what you can see here are hot springs. Yellowstone is basically the residue of a volcano that erupted many, many years ago, but it is still quite active. There are still a lot of thermal vents coming up out of the Earth. The water there is usually very warm. And one can see here a variety of organisms that grow. And in this case, I'm just showing and focusing on some of the phototrophs that sit in the hot spring outflow. Inside are non-phototrophic organisms. These are organisms that take their nutrients from the minerals that are present in the pools, in the crater pools of Yellowstone. Something else that you also find in Yellowstone are these sulfur-rich hot springs. These are just really beds of sulfur that are coming up out of the Earth. And surprisingly, even here, where the temperature is often quite hot, our organisms called Sulfur Lobus, which are archaea, not bacteria, they're archaea, but again, single-celled organisms, and live in abundance. And in fact, it is true that everywhere you go on Earth, you will find that there are microorganisms. And these microorganisms sustain the environment, they make the environment work, and in particular, all of the animals and all of the plants that are present on Earth require and depend upon these microorganisms. Now, I'm going to move now to a more specialized area and talk about the human microorganisms. We call them the microbiome. These are the organisms that live with us and upon which we depend for our lives. On the skin, our skin is actually crawling with bacteria. Staphylococcus, Corynebacterium, Propionobacterium, etc. Lots and lots of organisms. And most of these are harmless. Most of these do not do any harm to us, but rather they keep our skin in good condition. In the mouth, we have Streptococcus, Lactobacillus, Fusobacteria, organisms that help us digest food and that keep our mouths healthy. Of course, some of them do have some problems when they affect our teeth. In the respiratory tract, we have Streptococcus, Staphylococcus, Corynebacterium. In the gastrointestinal tract, we're leading down into our stomachs, many organisms that are absolutely essential for our ability to metabolize the food that we eat. And finally, in the urogenital tract, there are also many organisms. Some of these, again, can cause problems, but most of them are harmless. And I think this is the message that I really want you to take from this talk. And that is that bacteria help us. Without bacteria, we run into all sorts of problems. If we were to find an antibiotic that would kill all of the bacteria that live with us, we would die. We cannot live without them. They are our friends, and we should be very careful about how we treat them. Now, of course, most bacteria that you have heard of and that most people hear about get a lot of bad publicity, and they do so because the microbiologists, starting in the late 19th century, started to focus on those bacteria that cause diseases. This, of course, was of interest to doctors who particularly wanted to know how you stop disease, but they also became the focus of research in this area, the focus of the bacteriologists and the geneticists. And as a result, the journalists and the news media eventually focused on them too. And that is why they ended up getting a lot of bad publicity. We only heard about the disease-causing organisms. We heard very little about the good bacteria and those that were necessary to keep us alive. And if you look down the list of disease, you will see many that you have heard of: anthrax, gonorrhea, typhoid fever, diarrhea, tetanus, botulism, dysentery, syphilis, whooping cough, all diseases that none of us want to get, that some 100, 150 years ago, these were major sources of death for humans. And slowly we learned they were present, we learned how to control them. Along came the antibiotics, and along came various ways of dealing with these bacteria. I want to take you through and talk about a few of them, because some of them are actually rather interesting. They have interesting properties, even though they cause disease. And as you will see, not all of them are really that dangerous. I want to start off with Helicobacter pylori. I'm actually engaged in a big project with the National Cancer Institute at the moment, in which we're looking at Helicobacter pylori, which lives in the stomach and has been associated with gastric cancer. This particular bacterium can bind to the epithelium in the stomach, the lining of the stomach, and can inject its DNAs into cells in the stomach and cause gastric cancer. Not all Helicobacter does this, but quite a lot do, and we don't know why or how cells become cancerous as a result of their interaction with Helicobacter. But all the news about Helicobacter is not bad. It turns out that for many people, Helicobacter are actually beneficial because they stop other more dangerous bacteria from growing. And so, as is so often the case when we're starting to look at human disease, we discover that one of the problems is that the disease-causing bacteria actually do have some good properties when they're not causing disease. And for the most part, the good bacteria that live with us actually prevent the bad bacteria from growing. So many of our bacteria help us by keeping disease bacteria away. This is something that you've probably not come across in India. I've not heard of cases in India, but maybe they exist. But it's certainly very true in the part of the US where I live, is a disease called Lyme disease. It's a disease that is characterized by being bitten by a tick and then generating the lesions shown on this slide. But they don't last for very long. They maybe just last for a few days or a week, and then they disappear. But the bacterium that causes them, Borrelia, in particular Borrelia burgdorferi, is a spirochete bacterium, and it continues to live. It lives within our bodies once it's got in, and if it gets into the nervous system, can cause a lot of problems. It's also a bacterium that is not that easy to kill. There are bacterial antibiotics, rather, that will defeat Borrelia, but they take a long time to act. And so sometimes you have to take antibiotics for maybe 4, 5, 6 months before you get rid of all of the Borrelia. And our natural bacteria don't seem to be able to deal with this so terribly well. Now, another disease that I'm sure you've heard of is cholera. Cholera often accompanies getting bad water into the water system and getting bacteria in. And it's caused by an organism called Vibrio cholerae. And this is shown here. You can see the Vibrio at the bottom of the picture, all the small things. But it's rather interesting that Vibrio doesn't usually like to live by itself. Rather, it lives next to and sometimes binding to a eukaryotic organism called Volvox. And interestingly, whenever one has cholera infections, there is actually a very easy way of getting rid of the Vibrio in the water if you happen to live in India or in Bangladesh or in that part of the world, where the ladies wear very tight woven saris. And Rita Colwell, who used to be the head of the National Science Foundation, discovered that if you take water that carries Vibrio cholerae and just filter it through a very tight woven sari, you get rid of the Vibrio. And you do so because the Vibrio binds to the Volvox, and the Volvox cannot get through the sari. The thread is so fine and the weaving is so fine, it completely stops this. And so this has turned out to be very useful in poor communities, which typically didn't have access to antibiotics and often ran into problems with their water. So the saris turn out to be very interesting and useful. Another disease that you may have heard of is called botulinus. And botulinum toxin is not just bad for you, but interestingly, it's also something that ladies in the developed world often deliberately inject into themselves, particularly into their lips, to make their lips larger. Clostridium is caused, Clostridium botulinum is the product of, is this gene, this organism here shown flying around in the water. And Clostridium botulinum and all Clostridia have an interesting property. It turns out that when you try to treat them with antibiotics, they do something called sporulation. They form spores, little organs that are not growing, that contain all of the genetic material for Clostridium, and then they sit and they wait until the antibiotic goes away, and then they start growing again. And this is really something that is very interesting, and something that is rather interesting to me personally, because it turns out the signal that causes Clostridium to sporulate is caused by a DNA methyltransferase, an enzyme that puts a methyl group onto DNA, and in this way triggers the genes necessary to form spores. And my own personal interest is in DNA methylation. And when we discovered this methylase, it seemed clear that here was an opportunity to find a drug that could disable this methyltransferase, and in this way stop Clostridium from sporulating, and therefore perhaps help get rid of clostridial disease. Some clinical trials aimed at this are underway at the moment, but we don't have any results yet, or at least none that I've heard of. Another nasty disease is typhus, and typhus is caused by organisms called rickettsia. And these are bacteria that don't actually live outside of the cell, but rather they're intracellular. They live inside human cells. And that, of course, can make them very difficult to get rid of sometimes. Here's another rather interesting and nice looking spiral bacteria called Treponema denticola. And this is something that you find in the mouth, and that if you're not careful, this can get into your teeth, and is probably not something that you particularly want to get involved with if you can avoid it, but a rather nice looking bacteria under the microscope. Now here's an organism, Yesinia pestis, which is the agent that caused massive disease in Europe in the Middle Ages. This is the cause of the plague, which wiped out 90% of the population of Europe in the Middle Ages. It's an organism that we now know how to deal with. It's sensitive to antibiotics, and yet still we have problems with Yesinia. It is present in the environment. It is carried by rats, and in many parts of the US and elsewhere, Yesinia shows up quite often. But usually, if you're living in Arizona or New Mexico or the Western states where this does show up from time to time, doctors know exactly what to do, give you an antibiotic, and away it goes. However, very often people who live in Arizona travel elsewhere. Maybe they go to New York City, maybe they go to Boston where I live. And when they come down with the symptoms of this disease, the doctors have never seen it before because it is not endemic where I live. And if it's not treated quickly, this can become a major problem. And you can see these buboes, these little growths that occur, and very often doctors will attribute these growths to things other than Yesinia pestis. And if you don't take an antibiotic quickly, Yesinia pestis can be very nasty. It can lead to this gangrenous nature. Here you see gangrene in the fingers, in the tips of the fingers, caused by Yesinia pestis. This is the organism itself, rather innocuous looking red cells that are sitting around. And yet this disease, this one organism, probably has done more damage over the course of history than almost any other bacterium that we can think of. Now, what I don't want you to do is to come away with the idea that we're just surrounded by nasty bacteria. We have some, but they are not predominant. They are not the major bacteria that we have living with us. Rather, they are an aberration, if you will. Because bacteria are absolutely essential for our health. They protect us from pathogens. They protect us from cancer. And the way to think of it is that they have made us their home. They want us to be well. They want us to be safe. They don't want anything bad to happen to their home. And so they protect us. And the kinds of protection that they provide is a very interesting one, in that they have found ways to stop other bacteria, and in particular, a lot of pathogenic bacteria, from growing. And in this way, they've actually provided very often a source of antibiotics by looking at the bacteria that normally live with us and looking at the compounds and proteins that they make. In this way, we can find how they do it, how they keep the bad guys from growing, and then we can use that knowledge in order to develop pharmaceuticals that will help us too. But you have to realize that this is an arms race. The pathogens and the good bacteria are always fighting with one another. They don't want to give up that fight, so the pathogens are constantly finding ways to evade antibiotics, to evade the natural compounds that the good bacteria provide, and so on. And something that's happened relatively recently, and which was not known for a long time, is that not only do bacteria protect us from other predatory bacteria, the so-called pathogens, they can also help us against cancer. And it's recently been found that there are a number of bacteria that actually stop cancerous cells from growing. In effect, they're a surveillance system. They wander around our bodies. If they find cells becoming cancerous, then they can stop those cancerous cells. We don't yet know how they do that. We want to find out, of course, but that also leads to the possibility that there are many other things that they perhaps do that are good and that we don't know about. And you have to realize that studies of the good bacteria, of the metagenome, as we call it, the microbiome that live with us, studies of these organisms are still in their infancies. Until relatively recently, maybe 10, 15 years ago, we knew that these bacteria existed, but no one studied them because everybody was so busy studying the pathogens. But now, a lot of laboratories, a lot of bacterial laboratories, genomic laboratories around the world are actually looking at the microbiome. They're trying to work out just what it is that these microorganisms are able to do to protect us, not just to keep themselves safe, but also to protect us. I want to talk a little bit about probiotics. I'm sure this is something that's very familiar, particularly in India, because I think a lot of Ayurvedic medicine and a lot of medical practices in India and in many of the developing countries depend upon probiotics, depend upon compounds that one can introduce that can help keep people healthy, and also become aware that organisms like lactobacillus and bifidobacteria, which are now being very heavily studied, these also are organisms that can help protect us against pathogens. And as I mentioned previously, Helicobacter is one such bacterium. Even though Helicobacter can cause gastric cancer, can also cause ulcers, and it can have other deleterious consequences. If we completely eradicate Helicobacter, other bacteria grow, and that can be even more dangerous. How many genera of bacteria are they that operate in this way? Well, I put 5 question marks at the bottom. I probably should have put 5,000 question marks at the bottom, because the bottom line is we do not know. This is an area of very burgeoning research. It's an area that we're anxious to find out more about, and many labs are studying these organisms that live with us. They are our friends, and these probiotic bacteria are especially our friends. Here, non-fat yogurt. Yogurt is full of lactobacilli. Sometimes it also has other bifidobacteria and others in it, but it tends to be fairly healthy. And there are an awful lot of people who depend quite heavily on compounds that are present in yogurts and other foods that really make it very good for us to eat them and to maintain a healthy lifestyle. Food can be just incredibly important, and it is, I think, not unknown in India that the quality and the types of food that we eat can make a huge difference to our lifestyles. And one of the problems that we find in the developing world, for instance, is that much of the food that is eaten, the so-called fast food, is actually not very healthy, is not good for you, and leads to obesity. This is something that you find in yogurt, Lactobacillus sakei. And I want to tell you a little story about lactobacillus, because it turns out that it is possible by genetic engineering using modern biotechnology techniques to actually improve the ability of lactobacillus to serve as a probiotic. Company Nestlé's were working in Switzerland, they're based in Switzerland, and they were doing some experiments with lactobacillus and just making genetic modifications of one sort and another, and came up with one, a Lactobacillus johnsonii strain, that was much better and much healthier for you than the natural Lactobacillus sakei. They were thinking of putting this into yogurt because it turns out they own a yogurt company. But then something unfortunate happened. In Switzerland, as in the rest of Europe, there was great resentment and great worry about genetically modified organisms, so-called GMOs. And for reasons that have more to do with politics and money than they do with science, GMOs became taboo within Europe, to a point where Nestlé's was so concerned that they were trying to improve yogurt that they stopped doing it. They were afraid that there would be demonstrations that would close the company down. And so they stopped their work. And I think this is a very unfortunate example of the way in which anti-GMO movements have really caused problems, not just in Europe, but in the rest of the world. If you recall the improvements in agriculture that have taken place in the West, in Europe, in the US, in Japan, and elsewhere, most of these are focused on foods that are widely eaten in the West, not necessarily on foods that are staples elsewhere. As a result of that, using traditional methods of breeding, it's been possible to really improve foods, improve crops to a point where you can get very high yields, you can stop pests from attacking them, and so on. But in the developing world, the same is not true. The big agricultural companies saw no way to make money by improving cassava, by improving cowpeas, by improving the crops that were widely eaten in the developing world. And as a result, very little improvement has taken place. And yet from our experience in the West, we know that it is possible to greatly improve yields, to greatly improve pest resistance, and so on. All of this can be done using GMO methods. The fact that Europe is opposed to it, not only has it affected Nestlé's and affected the improvements of foods in Europe, it is having a devastating effect in the developing world, because much of what they grow is exported to Europe, and Europe won't take genetically modified foods, except when it comes to feeding their cattle and feeding their sheep. And there they can import large amounts of GMO foods. Something a little hypocritical there about this. But I think this example of lactobacillus and yogurt is a good example of how anti-GMO, anti-science approaches are starting to cause problems. And as the climate warms, we will need GMO solutions to all sorts of agricultural problems. And if they're not allowed to take place, then another means of correcting climate change, of mitigating it, will be taken away from us. I want to switch for a moment and talk about Clostridium difficile. Clostridium difficile is a nasty organism. It causes colitis. It doesn't kill you, but it causes very unpleasant symptoms. And it is very difficult to get rid of, in part because it's a Clostridium and can form spores, and the spores are resistant to antibiotics. And as a result, if you now go into a hospital, there's a very good chance that you will pick up a case of Clostridium difficile. You will be given antibiotics, but the chances are that you're not going to recover from it. And so you will end up with a Clostridium difficile infection that cannot be treated. Now, you go home, you don't die of it, okay? It's just unpleasant, but the unpleasantness is that it causes diarrhea. And it causes unexpected diarrhea. You never know when it's going to happen. And so patients are sent home, but typically they will never leave their home again because they're afraid that they will have a sudden attack of diarrhea if they're on a bus or if they're out walking or in some place where it would be extremely embarrassing. That is a problem, and Clostridium difficile is a problem. But as I told you earlier, there is a potential solution that we know about. And this solution depends upon the fact that because this organism forms spores, and there is a DNA methylase that triggers this spore formation, if we can develop the correct kind of antibiotic, then we should be able to stop it. There is also another interesting way of curing C. difficile, and that is by something that we call a fecal transplant. Now, fecal transplant, you, I'm sure, know what feces are. In common vernacular, we call it shit. And in order to do a fecal transplant, doctors will collect feces from patients who are healthy. And healthy feces have within them something that will attack Clostridium difficile. And in fact, in many, many cases, in fact, in most cases, it is found that fecal transplants will cure a patient of Clostridium difficile. Something about the bacteria that are present in feces of healthy individuals can attack Clostridium. Now, there is a way of dealing with this, something called casu-gel capsules, among others. There are many other ways of doing this. These are basically capsules that contain extracts from feces. They are really very good. They dissolve slowly after you take them, and they really do a wonderful job in getting rid of C. difficile. Now I show the following picture, thanks to Elizabeth Homan, Betty Homan, who is at Mass General Hospital, who gave it to me. And this is intended to be funny. Unfortunately, the first type of capsules they came up with were translucent. You could see right through them. And although they were new and improved, they were acid resistant, they minimized the risk of a bad aftertaste. Nevertheless, people might have been unwilling to take them. So even sometimes good medicines can have a downside. This is really a joke. It's very easy to deal with this. They don't look like that. But it's kind of interesting that this idea of fecal transplants, of taking feces from healthy people and allowing the bacteria that were present in the feces to take over the microbiome of an unhealthy patient, can have health consequences and can improve the health of people, I think is something that is very interesting. We would like to know why. You don't really want to take whole feces. You would like to find out what are the good organisms and then make a cocktail of the good organisms and use those in order to treat people who have diseases where fecal transplants can help. Now, I want to finish this talk by showing you two slides. The first one is a picture of Mars. And as I'm sure you know, we have been doing a lot of exploration on Mars. And in particular, we have been looking to see if there is any signs of life on Mars. Now, we don't expect to find human life. We don't expect to find anything terribly sophisticated, but it is reasonable to think that there might well be bacteria or archaea that live on Mars, maybe not on the surface, maybe underground. And for me, one of the wishes before I die is that we find out if there is life on Mars, and if there is it older than life on Earth? Because it's always possible that life did not develop first on Earth, but actually developed on Mars and then went underground. And if any life on Mars was older than life on Earth, then that would mean that we are really all Martians. And that, I think, would be a wonderful fitting tribute to make us to really get put in our place and make us realize that life is really more complicated than we think. And the last thing I want to do is just to press the GMO case again. I've been organizing a petition by Nobel laureates. We now have 150 Nobel laureates who have signed up. We sent an open letter to Greenpeace and to every UN ambassador, urging them to look at the science, realize that GMOs are safe. There is not a single example of GMOs ever having caused a problem. And I rather like this cartoon on the right, which came from a newspaper in Iowa, in which the opponents of GMOs, the Greenpeaces of this world, are busy telling people you've got to be very dangerous. Do you know what this stuff can do to you? And the little boy in Africa or any developing country who is starving says, yes, he knows what it can do. It can keep him alive. And as I say, there is just no evidence, zero evidence, that GM foods are dangerous. No evidence that GM crops are dangerous, despite the massive campaigns that have been put out. And so I would urge you all to take a look at this website, the Nobel website, supportprecisionagriculture.org, which will list the names of the Nobel laureates who've signed on to this campaign and has a lot of information about GMOs and about the ways in which GMOs can be helpful, but particularly for the developing world. In the developed world, while we can benefit from them, we don't really need them. But in the developing world, countries like India, countries in Africa, they do need them. You do need GMOs. And it is very important that politicians learn just how valuable these can be in terms of keeping people alive. So with that, I'll close. I will be happy to take any questions that might come. And thank you all for listening.
Thank you, sir. Yeah, we have quite a few questions in the chat box. I'll read them for you.
Absolutely.
Some of the major questions go like this. Do antibiotics kill off the probiotics? And is it possible in the future to make antibiotics which are probiotic friendly?
So the answer to question one is yes. Antibiotics tend to target specific kinds of bacteria very often, but they don't distinguish always between the good and the bad. And this is why we run into all sorts of problems, because very often when you take antibiotics, you have all sorts of problems caused because you kill off the good bacteria too. And so it can sometimes take a while to get away from this. Now, there are some antibiotics that are more specific, but most of them are not that specific. Most of them have quite general bad properties in terms of killing bacteria. And while it's nice to get rid of the pathogens, and you know that eventually the good ones will come back, it's unfortunate that they kill them so prolifically to start off with. And so if you can avoid using antibiotics, if you can get better some other way, do so. And it has become a problem that doctors frequently employ antibiotics in situations where they're just not needed. You know, the mother shows up with the doctor, has a child who's got a sniffly disease, probably a viral infection, which antibiotics have no effect on whatsoever. But they say, oh, doctor, please prescribe me an antibiotic. In many countries, you don't even need to go to a doctor. You just go to the drugstore and you can buy them immediately. And this is not good. This is not good. One should use antibiotics very carefully because, like I say, most of the bacteria are good. They're trying to keep us well. Why would we want to kill them?
Yes. Yeah, another question is, can bacteria help fix the problem of non-biodegradable waste like plastic, say?
Sorry, can you say that again?
Yeah, can bacteria help fix the problem of non-biodegradable waste? Is there any bacteria that can feed on plastic?
Well, there are bacteria that can eat plastic. They don't do it very well. And there are some that have been studied quite extensively now in Japan. And they were discovered quite by accident. People are now looking much more widely to see if there are bacteria that can eat plastic. And perhaps the most exciting thing I've heard about recently is that there is a great deal of life associated with this huge collection of plastic in the Pacific Ocean. And we don't know much about the life, but it looks as though it is living off the plastic in some way. And if we can find out how the bacteria do that, then I think there is a possibility for really doing something about existing plastics. But for me, the most exciting prospect is that we can make plastics now that are biodegradable. We can make plastics that bacteria can eat. And I think the sooner we shift the plastic industry into making plastics that are recyclable, the better, because there's no doubt that the plastics we've produced so far are a major problem. They just get everywhere. They don't go away so easily.
Yes, sir. The next question is there any type of immune system that exists in bacteria?
Well, yes, actually, you know, bacteria have a big problem with things called phages. These are viruses that really grow on bacteria. And so the reason I first got interested in bacteria was mainly because of one of these immune systems. It's called a restriction modification system. It involves an enzyme that will cut DNA that is not methylated. Most phage DNA is not methylated, and so when the phage enters and attacks the bacterial cell, these systems will cut up the phage DNA and stop it from infecting the bacterium. There are many, many of these known, and in fact, the enzymes that cut DNA were the enzymes that allowed the biotechnology industry to take off in the 1970s. And quite a lot of these enzymes were discovered in my laboratory at Cold Spring Harbor at the time. We now know of even better ways of dealing with DNA, but still these restriction enzymes have turned out to be very useful. But we now also know that bacteria have many different ways of providing immunity against phage. And something like 10 or 12 have now been documented. There are a number of reviews out about this, and most of these operate in particular kinds of bacteria. And one that you may have heard of is the so-called CRISPR system, which is the way that modern biotechnologists start to try to change genomes. So these have become very useful for genetic engineering. And in fact, the Nobel Prize, not this year, last year in medicine, was awarded to Jennifer Doudna and to Emmanuel Charpentier for their discovery of CRISPRs and the fact you could use them for gene editing. And they are just another form of bacterial immune system.
Yeah, next, we'll take a question from Tamina. Tamina, can you unmute and ask so directly? Is Tamina there? Maybe I'll put the next question. So you talked about use of bacteria for treating cancer. And what's the extent of research or practice that is going in this field? How much is
okay. Yeah, so I know of preliminary results that have come out from the University of Chicago. There's a group at Chicago that have been doing this. And I know that as soon as that came out, there were a number of other groups that started to look and see if they could work out whether there were bacteria attacking cancer cells. But I don't know a great deal about this. This is not an area that I've followed terribly closely. But I do know that the preliminary results really looked very interesting. But I think you have to go and read the literature to find out more about that. I'm not an expert on that aspect of things.
Is Tamina over there? She has got a big question. Maybe she can ask it directly. So I'll read it out for you. She asked whether administering live microbes like probiotics, is it really helpful? And she's asking if we can administer cell-free nanoparticles. It will be more beneficial than putting in the live bacteria in your body.
Well, a lot there depends upon what is in the nanoparticle. So I think the problem there is that, you know, nanoparticles can be useful. The mRNA vaccines use nanoparticles to put the mRNAs in. And I'm sure there are other things that one can deliver via nanoparticles. But in general, for good probiotic bacteria, it is the bacteria themselves that are important, because the bacteria grow, they excrete compounds that will then make sure the environment is safe for you, safe for them. And so it's the growth of the bacteria that is important. And there are many ways in which you can introduce these. Nanoparticles would be one. You can put a bacterium inside a nanoparticle. But in general, you don't need to do that. There are many ways. You can just put them in solution and drink them, and that works quite well. Yogurt works quite well. There are many ways you can just add them to food and to then get them into the patient that way. Looks as though Tamara now is live.
Thank you so much, sir, for your answer. I would like to ask a follow-up question for that. So I also mentioned that using probiotics has several risks associated with it, and many cases have been reported so far all over the world related to deaths, allergic reactions, arrhythmia, etc. So that's why I asked that question in particular, because there are risks associated with administering probiotics. So the follow-up question is whether you mentioned about bacteriophages, and I'm particularly working in the field of bacteriophages. So my question is whether probiotic organisms have an influence of these bacteriophages, whether in the gut microbiome or as dormant phages that are integrated into their genome.
I think on the whole, the answer to that is that they will also be susceptible to phages unless, you know, they have suitable restriction systems or other systems to stop them. Most lactobacilli and bifidobacteria do have good immune systems. They don't become susceptible to phages. And there's no reason to think that they have an impact on the ability of other bacteria that are in your gut to suddenly become susceptible to phages. I don't know ever any work that would suggest that. I think they would remain, whatever was their natural susceptibility, that would remain. I don't think the probiotic bacteria would have much effect on that, if any.
Thank you so much. I really appreciate it.
Okay. You're welcome.
Yes, and the question is, can we treat a disease, a bacteria, with its same kind?
Sorry, say that again.
Can we treat a disease caused by a bacteria using the same kind of bacteria?
Well, yes, of course you can.
Mm-hmm.
I mean, with genetic engineering, you can introduce many genes that you might want to put into them. And if you wanted to make something dangerous, you could. But, you know, people always say, well, what about the terrorists? Are they going to try to make bad bacteria to kill us all? And I say you will usually find such terrorists because they're dying themselves, because they've made these bacteria and not taken the proper precautions to protect themselves from them. But I think it's generally true that most nations do not think biological weapons of this sort are ever going to be a problem. There are, you know, you just can't protect your own people against them. And so if you try to start such programs, you're going to cause widespread trouble everywhere, but including in your own country, and probably in your own country first. So, and that was the basis of these negotiations and the implementations of restrictions on the development of biological weapons. I think pretty much all countries now have signed on and said, this is not something they're interested in doing or want to do. They've closed down the facilities that maybe 50, 60 years ago were trying to form biological weapons.
So another question is, what's the limitation in studying bacteria of different kinds? Is it the culture conditions for that particular bacteria or anything else?
Yeah, so culture conditions can be a big problem, and especially when you're looking in the metagenome, because frequently you find that 3, 4, 5, maybe as many 10 bacteria all need to be present in order for the population to grow. That individual bacteria sometimes can't grow by themselves. They need these friends to grow with them. But very often, you find that you can provide the nutrients that the friends provide by putting a membrane down and taking an individual bacterium, putting it on top of a membrane, and then underneath the bacterium, you put the rest of the metagenome, and that the compounds produced by the metagenome will pass through the membrane and allow the individual to grow. And so there are quite a number of bacteriologists have made really good on this. I know one in particular at Northeastern University, Kim Lewis, who's been very successful in growing all sorts of individual organisms from metagenomes using this kind of approach. And there are others who use similar approaches. And so it's usually a question of finding what it is an individual bacterium needs in order to grow. And then if you can provide it by whatever means, then you can do it. But it can take a long time to find just what it is you need to add in order to have it to grow. And so this, of course, does make it difficult to study. But we can study populations of bacteria, we can study mixed cultures, and we can find out quite a lot about them. And the most important thing is we can sequence their genomes. And so once you have, now you know what it is, what it takes for that organism to grow. You know all the genes. We often don't know what the genes do. And so that's a whole nother area of research to work out what the genes do. And really, apart from hoping that we find life on Mars is older than life on Earth, I'm also hoping before I die that we will understand how a bacterium works, that we will understand the genes and the genetic makeup and the interactions to a point where we could write a computer program that would predict exactly how this bacterium is going to behave. So that's what I mean by knowing how it works.
Thank you, sir. I think we have come to an end for the question and answer session. Professor,
Yeah.
Professor Nitin, can I make just two comments?
Yeah.
One is that I remember with gratitude Professor Tony Leggett, and request Professor Roberts to say a big hello to Professor Tony Leggett in our name. I remember he was a previous speaker. And then also one quick comment about this fecal transplant. I'm happy to observe that, you know, this fecal transplant for liver cirrhosis is very successfully practiced in Kerala, in one of the hospitals here.
Good.
That's wonderful to know. Thank you.
Okay, well, thank you. And I'm just sorry I couldn't show up in person because Kerala is a part of India that I like very much. I've only been there once, but I really enjoyed my visit. So I was a little sad that I could not do this in person.
Maybe next time.
Okay. Thank you, Dr. Philip Leto, for facilitating the Q&A session, and special thanks to Sir Richard for your generosity in answering the questions.
Okay.
I invite Dr. Liju Jacob, Department of Biotechnology, to do the vote of thanks.
Good evening, everyone. St. Berchmans College is celebrating its centenary, and this is the second Nobel lecture of the year. It's my privilege and honor to propose a vote of thanks on this memorable occasion. St. Berchmans College is fortunate to have Sir Richard John Roberts deliver the 5th Berchmans Erudite Lecture. Your talk unraveled the fascinating world of bacteria, of which many are important to sustain human life and keep us alive and healthy. Thank you, sir, for your words of wisdom. It was an enriching and enjoyable experience listening to you. Now we understand, appreciate, and love bacteria better. Yes, sir. On behalf of St. Berchmans College community and all the participants, let me extend the most sincere gratitude for being with us and gracing this occasion with your inspiring lecture. Thank you very much, sir.
Okay. Thank you. It's been my pleasure.
On behalf of BELS, I would like to thank our manager, Reverend Father Thomas Pardiet, Principal Reverend Father Reggie Piquerian, Vice Principals, and the management for the immense support and guidance. Thanks to all the participants for your presence and interactions, which made the session lively and meaningful. I would also like to thank the Staff Association of St. Berchmans College for their support and encouragement. A big thanks to Professor P.J. Thomas, convener of the Berchman Erudite Lecture Series, and his team, Dr. Philip Leto Thomas and his team for coordinating the Q&A session, Master of Ceremony, Professor Nitin Verghese, the technical support team, and all the faculty members, especially from the departments of botany, zoology, biotechnology, and microbiology, for helping us organize this lecture. Once again, I thank each one of you for making this event a memorable one. Thank you.
So, thank you, Professor Roberts again.
So thank you for being here today, and I wish you all
Yeah.
a good evening. Bye. Thank you.
Bye.
Hope to see you in Kerala.